More than 50 scientists working on the WATBIO project gathered at the Venice International University Centre on the island of San Servolo for the consortium's second annual meeting in October 2014. The meeting was held over 2 days and included 9 detailed presentations of research progress. The participants in WATBIO ranged from those working for the first time in international research through to experienced scientists who've worked for many years together on poplar, miscanthus and arundo. WATBIO will run for 5 years. This meeting comes at the end of the project's second year. It is an important phase when the early results are emerging and when the detail of later stages of the research is still being planned. The project coordinator, Professor Gail Taylor of the University of Southampton in the United Kingdom, reflects on progress here.
I'm Professor Gail Taylor. I coordinate the Framework 7 European project WATBIO. The project's about bringing together new crops for bioenergy applications in a future drought-prone Europe. And it's about harnessing the new technologies in DNA sequencing to accelerate breeding and deployment of improved crop material. And in this new world we find ourselves where we can sequence virtually everything very, very cheaply, we can move beyond the concept of genetic modification and work on these really difficult traits where we can improve plants and provide this material for a future Europe. Well, we've been running for 24 months. We've got together this big partnership. It's 22 partners, and 7 of the partners are from industry or commerce, and they all have a rather different role in the supply of these biofuel crops. And so we've brought the partnership together, and we're from rather different disciplines. So physiologists, ecophysiologists, crop breeders, geneticists, people interested in genomes and DNA sequencing. So we're working together in a very productive way, and we've achieved a number of things in the first 24 months. For example, we've cohered a very vast number of unique genetic resources in poplar, in arundo and in miscanthus. So we've planted 12,000 poplar trees, we've planted several hundred miscanthus, and we've brought together a brand new collection of arundo. So bringing the collections together has been one achievement. Now, aligned to that, we've also sequenced several million sequence reads of these plants, and we're looking through the data to look for unique gene expression patterns and to provide genotyping tools which can accelerate breeding in future. So we've achieved quite a lot, and we're looking forward to the remaining 3 years of the project. It's a great challenge to breed these new crops. What I would say is, though, that this project, it differs in a couple of ways to what's gone on in the past. The first is that these bioenergy crops are not reliant on grain filling. So we're just interested in the production of raw vegetative biomass, very fast growing plants that develop biomass quickly. And so that's a slightly easier thing to resolve at the level of the genome. So that's the first thing I would say. And then the second thing is, in history, plant breeding has been done very traditionally, but that was before we had this next generation DNA sequencing, where we can literally, for €1,000, gain an incredible amount of genomic information from a particular plant. And that puts us in an entirely different paradigm to how we can breed in the future. So I would say that things are looking optimistic for the future, that we can accelerate those breeding routes that many of us are familiar with, that have taken a long time in the past. They do. They take a long time with trees, largely because trees don't flower very often, or the flowering takes a long time. The crosses are difficult to make, and they're very outbreeding. So we have those issues with trees. But on the other hand, the trees we work with, poplar, have some massive advantage because they can be propagated vegetatively or clonally. So we can make what we call immortal plants. So if there's a particular tree that I'm interested in, or 1,000 trees in my case, we can propagate them, put them in several environments, and begin to untangle this complex genotype times environment interaction. And so it can actually be an advantage that we work on these long-lived trees.